Hello, everybody. Thanks for the invitation. It's wonderful to come and talk to people about our science. Been really enjoying our trip to Chile. Started with Rapa Nui, and then we went to Santiago and Valparaiso, and now here. And looking forward to go to Alma Observatory afterwards. So thank you for all the great hospitality. So today, are you ready for some science? I tried to make it less technical, and hopefully there'll be some time afterwards for questions and answers. And I know this is being translated into Spanish in some of your ears, so I'll try to talk slowly. It's good to start with always giving credit, and this is a picture of my lab, the people, the students, PhD students, postdocs who do the work. And this is a few years ago when we had a reunion. So that means my lab is not this big. So we have current trainees as well as alumni who came back, and we had a day of science. And I owe it all to these young researchers for the work that I will be describing to you. So we study sensory biology. So how do you see, smell, feel a rose, for example? And ever since the days of Aristotle, the senses were divided into 5 senses. So you have olfaction, which is smell. And here, for example, just coffee has about thousands of compounds that are in the air that you smell, and together that comes to your nose and you understand it as, morning coffee. The point is both that and taste, which is simpler, just sweet, sour, umami, bitter, etc., is all chemicals. These are chemicals getting into your nose and mouth to get that sensation. Your vision, sight, is based on physics, so you have to translate photons into chemical or electrical energy that your neurons will understand. There's also hearing, which is mechanical. It's all sensing pressure in your ears. But the most interesting sense, if you ask me, is the sense of touch. People have described it as very profound. It's complex because, unlike the other senses, which is very localized, for example, the eyes, only through your eyes, your sense of touch is distributed all over your body. In addition, it has many different modalities. So touch can mean temperature, can mean pressure. If you consider itch, you would also consider chemicals that causes this somatosensation. So temperature, for example, is very important to us. There's a very narrow range of temperature in which we can survive. And because of this, and our civilization, we have become very particular to what temperatures we like. And whenever that happens, businesses take advantage of this. Now, modern cars have dual temperature control so that the driver and the passenger will enjoy different temperatures. We all have our specific preferences for temperature. Also, if you enjoy wine, you know that red wine and white wine are stored and enjoyed at very different temperatures. So we're very picky about temperature. When we talk about touch, though, you mainly think of this pressure sensing, right? And our sense of pressure is remarkable in its sensitivity. I can tell you that we sense a few nanometers of indentation in your fingertips. To give you an idea, that is 1 in 500th diameter of a human hair. So the slightest indentation you can feel with your fingers. So quite amazing. On top of that, we rely on touch for much more than that, for our emotional health. A hug from a loved one is so precious to us, and that's initiated by the sense of touch. There's also this other sense related to touch that I'm sure most of you don't know about it, but I'm going to argue and come back to this, that it's perhaps the most important sense that we don't know about, and it's called proprioception. So it's the sense of where your limbs are in space. If you think about it, I can close my eyes and I can bring my fingers and touch my nose. If you really think about how I did that without seeing, the only way is that you have sensors in your muscles, and from how much they are stretched, you know exactly where your hands are. So this is how you can do all these tasks. And as we shall see, they are very important for balance, for walking, for everything we take for granted. I'm happy to talk about later why we are not aware of this, why we don't talk about it. Partly it's because you can't turn it off. You can close your eyes and imagine a world without sight, but you can't turn off proprioception, so we take it for granted. But this represents the touch in better than any picture I know. This little boy is in the Oval Office of the White House, and he tells the president that his friends tell him that his hair looks like President Obama's. And Barack, of course, doesn't miss a beat, bends down and says, touch it. And so we often say seeing is believing, but clearly Barack knows that touching is believing too. And this is, of course, affects not just sensing this texture that I talked about, but again, that affective touch of touching the hair of the president for this little boy and realizing that someone that looks like him, that feels like him, is in the White House is very important. So the flip side of touch is pain, and acute pain is essential for survival. That's why it's evolved. But in many instances, we have chronic pain that is defined as pain that persists after the stimulus that caused it is gone. So it's like constant pain. And this is a major unmet medical need. If you followed pains, you know, the best drugs we have out there are opioids, and they have so many problems in addiction and side effects. For severe neuropathic pain, we don't have very good medication. So this is the application of what we're interested in, sense of touch and pain. So all of these touch, pain, temperature sensing start neurons that are on both sides of your spinal cord. And they're neurons, so they're cells, but they have thin wires, processes that go all over your body, and they're like antennas to monitor what's going on. And this video is a video from a zebrafish, another vertebrate, that you can see the bulbs are the groups of thousands of neurons that are extend to your body. And what the video will show is how they send these little neurites, the antenna, all over body as detectors of various stimuli that I talked about. So, you know, if you build a house and you want to wire it, you build the house and then know exactly where you want to put the wires. Animals have to do this difficult thing of, while they're building their body, they also have to build the sensory circuits to be needed later on to sense the environment. So it's a beautiful thing. So that was the real video. This is a schematic of a vertebrate. You can see the spinal cord in the middle, and those colorful neurons that I've labeled are in the dorsal root ganglia, these sensory neurons that sense all of these stimuli. And what they do is they gather information from the skin and muscle, for example, and bring it to the spinal cord and the brain by wires, just like electrical wiring. But they are very much specialized. So you have some in black sense temperature and pain, some proprioceptors sense muscle stretch in red, and in the green are many of the touch sensing neurons. So if we zoom in to where they go in the periphery, the arm is one example of a peripheral tissue, for example. You can see in red that the proprioceptors wrap around muscles. That's why I said they sense muscle stretch. While others extend into the skin, and I'm not going to go into details, but you have a variety of beautiful nerve endings in the skin that all together give you the signal that you think of as touch. We'll also talk a little bit about these sensors innervating internal organs, such as the bladder for sensing the stretch to tell you when you need to go to the restroom. One of the characteristics of these neurons is that they are extremely long. So they're very thin, microscopic, but because some neurons have to innervate your big toe, and that little wire has to come all the way to the neuron in the back, and then that has to send another wire to the brain, they could be 2 meters long, as illustrated by this basketball player. This is a simplified version of what these neurons do. In the skin, for example, they have to sense force and temperature and send this message to the brain. What we're really interested is in these proteins, the actual sensors, detectors, that initially feel the temperature, feel the pressure, and translate this to a language that neurons understand, which is electrical signaling. And these molecules are called ion channels. Ion channels, you can think of them as switches. So you go into a room, you switch the light on, and the light comes on. So that's the initial trigger to activate these neurons. What they do in the cell, every cell is surrounded by this lipid protective layer, and they sit right in the edge of the outside and the inside of the cell. And then when they're quote unquote open, ions flow in, and that's the electrical activity. So very simple signaling mechanism. They're remarkable proteins. When ion channels open, a single protein can let 10 million ions in per second. That's remarkable. And as I said, once activated, they change the voltage of the cell, which is the signal necessary to go from neuron to neuron and to the brain. So ion channels have been identified, and many are actually, you might not know their names, but you might take medicines that treat variety of diseases or conditions such as anxiety, arrhythmia, cystic fibrosis, epilepsy, some pain, stroke, and many of the anesthetics that you take before surgery, for example, affect ion channels. Again, I want to give you an idea of the scale of how small these ion channels are. So if you think of a grain of rice is 500 times bigger than a cell. So if you take that little and make it big, this is a cell. It's in the order of micrometers. Ion channels are in nanometer scale, which means on a cell, they would look like that little black dot. So we're talking really, really small. And so I told you many ion channels are already identified, but we are interested in something very unique that nobody had found before, which is we want ion channels that don't get activated by other chemicals. So for example, your brain is full of ion channels that respond to neurotransmitters like dopamine, etc. We are looking for ion channels that respond to temperature and pressure. These are physical forces, so very unique. So they were known to exist, but nobody knew what genes coded for these ion channels or proteins. So why do we even bother to study this? Well, honestly, the first reason is curiosity. Most of science is driven by just simple curiosity. And how do you change physical stimulus into chemical or ion gradients that your neurons can understand? I find very fascinating. And it's incredible to me that no one had found it till now. But of course, as I mentioned, there is relevance to human health in the sense of potential pain medication. And what's wonderful about science that you'll see in a minute is that you start asking one question and it leads you to something else. So there's always application that initially you didn't think about. Okay, this was kind of the introduction. Now I'm going to show you some real data, but again, I'll try to keep it simple. First, I have to tell you, how do we even measure from these micro, nano machines that, let's say, get activated by cold? So my lab identified this channel. The name is not important, TRPM8, but it is cold activated. So we have this technique called patch clamp. So in tiny cell, we, with the glass, grab onto a small part of the cell. You can see there's supposedly one channel, looks like a flower here. And electrically, we can actually record from this. Just like you can record electricity, we can record from a single protein. So in this example, in about 1 second, we have cooled the buffer around this from 30 degrees centigrade to 18 degrees. And what you're seeing, those little flickers, are the channel opening and closing. So initially, one channel opens one, and then in this patch, there's actually 3 or 4 channels, because at the end, you can see they're staying open. You have 3 or 4 of them. If you look at the y-axis, we mean this one, it's called picoamps, which is 10 to the power minus 12. I just wanted to get a scale of how small these currents are. 15 picoamps, which is, you see all those zeros? And 1 amp, if it was 1 instead of all those zeros, it would be the amount of current that one light bulb would have. So these are tiny currents, so to measure this, we have to have massive buffering and amplifying systems to be able to record them. Once we cloned this channel that's activated by cold, we found out something very interesting, that they also respond to the active ingredient in mints called menthol, which many of you are familiar with. because they are present in many over-the-counter products that give that cooling sensation. So for example, chewing gum, it's already called double mint. If you chewing it and then drink some cold water, you feel that rush of coldness. That's because menthol, this chemical, is activating your cold receptors. In studies that I'm not going to tell you, that we found that without tryptamate, animals can sense cold. So this is the way to sense cold temperatures. Another channel we found in a similar way that's activated by painfully cold temperatures. We also found that it responds to chemicals such as tryptamated. However, the sensation you get from it is very different. This channel feels like burning when you activate it. So I don't know if you have this in Chile, but there's these mouthwashes that you use in your mouth, and they make you believe that gargling that will kill bacteria. The sensation you're getting from it is simply because you're causing pain in your mouth. And so next time, if you take anything from this talk, just whenever you consume some of these products, remember, say, oh yeah, Ardem told us how you sense pain and cooling from these little mouthwashes and chewing gums. If you like sushi and you lose wasabi, again, that pungent burning sensation in the mouth and the nose is because of a chemical that activates tryptase-1. Even raw garlic has that burning sensation. When you cook it, it turns into another chemical that doesn't burn. So this is all tryptase-1. But what we find out because of these studies is that tryptase-1 is actually a very general sensor of pain. And there's lots of studies now suggest that if you block it, you might actually get interesting block of clinically relevant pain conditions as well as cough. And there are now small molecules, drug-like molecules by various pharmaceutical companies testing in human clinical trials if this is useful for treating pain. But what I want to tell you is how long it takes from the basic science that starts with a curiosity-driven work to potentially become medicines of the future. We identified tryptase-1 in 2003, and now we're 2025, so over 20 years to translate basic science into, hopefully, future medicines. So, apart from temperature, I also want to talk about pressure, but pressure is actually harder to study at the cellular level. For temperature, it's very easy. You cool the buffer, you warm the buffer. Here, we need to do something more specialized. And this is the schematic. You have that round thing is a cell. You're recording from it from the right. From the left, we come with a glass probe and actually push on the cell. So this is a video showing actually what's happening. This is the recording pipette. There's the cell, and we're actually pushing the cell. And if that cell has this ion channel that we're looking for that we didn't know, you would see this current and you can electrically record from them. So here I'm going to go a little bit into details about how we found this channel because I think it has some interesting general messages. We said if we can find a cell, easy to work with cell that has this activity, then we can make a list of genes. If someone doesn't know the molecular biology, simple. So you have 20,000 genes in your genome, and some of them are expressed in some cells. So you make RNA and then you make protein. So protein is the sensor we're looking for. But to do that, we need to know what genes can be this protein. Without getting into details, we found about 300 genes that could account for this activity in this cell. And so the idea is my postdoctoral fellow would knock out, delete one gene at a time, and see if this activity is still there. So I'm going to show you the data that gave us the molecule that caused me getting the Nobel Prize. And so what you're looking at is this current he's recording from this cell. And each of these is one gene being knocked down. So candidate 1, that first black one, he knocks it out. The current's still pretty big, so that's probably not it. He is very tenacious because each of these took him about 3 days to record to know for sure that this is not it, okay? So imagine for 11 months, he comes in every day, full-time works very hard, goes home, nothing, but didn't quit, didn't give up. the number 72 was this unknown gene called FAM38A. When he knocked it out, the current almost went down to nothing. So we got very, very exciting day. He renamed this piezo1 from the Greek piezi, meaning pressure. So in biology, we have terms that we call necessary and sufficient, very important. So necessary means it's required. So you take this gene out, you don't see this current. This is important, but it doesn't tell the whole story. We also want it to be sufficient, which means you take this protein, put it in a cell that's naive, doesn't have mechanosensitivity, and if you can record this current, it means it's also sufficient. And this is that experiment. So he takes a cell that usually doesn't respond to mechanical stimuli, and then he pushes them after putting piezo1 in. You can see this beautiful currents that he's recording, right? And this is quantitation, so control doesn't respond at all. You put piezo1, massive responses. So there's two piezos in your genome, two genes, two proteins, piezo1 and piezo2. They do about the same thing. So what do they look like? Proteins have a specific shape. I drew schematics of ion channels. Now I'll show you what they really look like. So I told you already that ion channels stay in the plasma membrane, right? In that fatty layer that surrounds the cell. Usually ion channels have 2 or 6 what we call transmembrane proteins, which means the protein weaves in and out of that lipid bilayer. This guy, piezos, had 38, which was more than any other protein that we had ever found. Now this is just a schematic. We actually can solve the structure of these ion channels, which means we know exactly what they look like. And we did this with other groups. And they come together, 3 of these 38 transmembranes, to form this monstrous but beautiful structures. So what you can see is each of these ribbons is one of these transmembrane domains. And each color is for a different of the 3 identical proteins that come together. And in the middle, which you can't see very well here, is the pore where ions go through. And if you can see, one of the attributes of this protein is it has this puckered shape. Kind of looks like it's bent upwards. And the idea has been that perhaps this is why they're pressure sensors. That if you pull on the membrane, they flatten and let ions go through. And now there's evidence that that's exactly what happens. So I'm going to show you morphing of 2 structures. This is when it's closed, bent like this. And then when you stretch it, it opens up magically. And this closing and opening is actually the basis of how they're pressure sensing. You put stretch on the membrane, they open, and they let ions in. This is one of my favorite slides. I love to show these molecular micro machines that have evolved to form this beautiful structure so that they immediately explain what their role is. But I love talking about piezos all the time, and I don't always have a slide to show. So a few months ago, I decided to get a tattoo of the structure on my body so that I can show it if I'm having lunch or at a bar. And I thought I'd show it to you because I put it in a very strategic place so that I can demonstrate the mechanism of activation. So this is closed, and then you open. So I can talk about my protein no matter where I am. Okay. So this is what it looks like in a membrane. So this blue is the plasma membrane, the lipid bilayer, and these guys sit, make little holes in it, and when membrane is stretched, they get activated. Pretty simple. So the rest of the talk, I'm going to go from the molecules to what they do in animals. We mainly study mice as an animal mouse model of humans, but we're also getting lots of clinical data from humans to find out what they really do. And loss of function, gain of function just means what happens if you delete the gene from the genome and what happens if you put too much of it. We're not going to talk about the too much of it, but that's one of the ways scientists use to find out what something does. You take it away and see what's missing, then you know if it's important for that. So for example, you want to know, is piezo, one of them, required for touch sensation, right? So we got very excited when we found one of the piezos, piezo 2, is expressed in these dorsal root ganglia, the neurons I was talking to you about, which account for touch, proprioception, etc. And this just shows in the cell bodies, which are next to your spinal cord, that the RNA, I told you DNA gives to RNA. So if you see RNA, it means it's on there, is expressed in many of the neurons of the RGs. And very interestingly, the protein, this is piezo protein actually looking at the fingertips where the nerve endings are. So the neuron starts here, comes all the way here, and the protein is exactly where it's supposed to be, where it's doing the sensing. So we made mice that don't have piezo 2. And so how do you ask if they sense touch or not? At first, we did some very simple experiments, and this we call tape on the back. So on the left is a normal wild-type animal. You put a tape on the back, they get really annoyed, just like you would if I did that to you, and they try to take it out. In the knockout, you can see already that they're not very coordinated, but they don't seem to care at all. You can do very different assays. This is one that you poke the limb, and you see when they lift it. If they sense it, they don't like it, they would lift it. Again, this is the wild type in gray. After the force increases, you can see more and more paw withdrawal. You can see almost nothing in the knockout. So these animals really don't sense touch. How about proprioception? Just to remind you, this is the sense of your muscles to know where your limbs are. The schematic on the left just shows what you can potentially do if you have very good proprioception and practice, that blindfolded you can do tightrope walking. The video on the right that I'll just show you, I just recently gave a talk at Davos, and before I talked, this musician came up and played a piece of classical music blindfolded. And the idea is that the only way he can do this is because of proprioception, because he can't see. And interestingly, this gentleman had never played this piece blindfolded before. He's a professional musician. He's played it all the time, but this was the first time he was going to play it blindfolded. Remarkable, isn't it? Okay, I just added this slide. This is right outside, right before I came in. Nancy, my wife, took this video. This also requires proprioception. This is a little game that you have to put that ring through that hole, and you have to, after trying a few times, you have to realize how much to push. All of this requires very fine hand coordinations. And thank you, thank you. What this doesn't show the 100 times I did and couldn't get it beforehand. So our collaborators at the National Institutes of Health actually identified people who do not have piezo 2. And remarkably, just like what we had seen in the mice, without piezo 2, these individuals don't sense touch, don't have proprioception. And one of the ways you can test proprioception is the same things as what Yuki, the violin player, was doing. But instead of playing violin, we're asking them to do something very simple, which is while blindfolded, just walk in a straight line. This is something that you and I can do easily, but she's harnessed to make sure she doesn't fall down. And as you will see, she has a very hard time performing this task. How about pain? There's no major deficits of acute pain, so hammer hitting your finger. So actually, there's another channel responsible for this that to this day we don't know what that is. But chronic pain, and one form of it, tactile allodynia, which means when touch becomes painful. It's actually really simple. For example, after sunburn or an injury, just touching your shoulders become very painful. This is not a major clinical condition. However, patients suffering from neuropathic pain experience this all the time, and there's no good medications for it. So we wanted to ask, is tactile allodynia affected in patients without piezo2 or in mice without piezo2. I won't show you the animal experiments, but my colleagues did this one experiment to demonstrate that tactile allodynia depends on piezo2. And to do this, they rub capsaicin on one part of the arm, which gets inflamed. You can see it becomes red, either in control individuals or in these individuals that don't have piezo2. And then you ask them if box A or B is painful. So for the control individuals, for you and me, of course, 100% of the time, you will know which side is painful. But for the patients, they don't feel anything at all. So you tell them that the way this test works, you have to pick A or B. They pick randomly and get it right 50% of the time. So that was important because now we know that blocking piezo2 will potentially be helpful in clinical settings for pain medication. And we are working on finding such molecules right now. But I just wanted to summarize what we found from these studies, both in mice and in humans, that piezo2 is the principal sensor for touch, proprioception, one form of pain, but also mechanical itch that I won't talk about. But in red, I also have this idea of interoception, which I introduced earlier, that it's the sensation of internal organs. And with collaborators, we have shown that many of these things that initially you don't think about that are pressure sensing are indeed dependent on piezos. So for example, with breathing, with every breath you take, your lungs expand, and you sense this through piezos and regulate your breathing pattern. Many of the patients without piezo2 actually go to the hospital because they have breathing problems, especially during their early years. Baroreception is sensing of blood pressure through your neurons. And we've shown that if you lack both piezo 1 and 2, you don't regulate your blood pressure very well. I mentioned urination. So the first two reflexes you're not aware of. You're not aware of sensing blood pressure. It just works in your unconscious. But other interoceptions you're very aware of because that's the whole point. And urination, again, every time your bladder is filling, you get the signal to go to the restroom. So these individuals without piezo2 don't have this sense. And so ever since they were kids, they've learned to go to the restroom 4 or 5 times a day to avoid accidents. Gut motility is another thing you're not aware of that your digestive system does all the time. When there's food going into intestine, mechanically you sense this and you slow down the motility of the gut so you digest it well. Again, piezo dependent. And there's many other things that we haven't looked at yet, but very exciting to look at in the future. If you eat a large meal, which I've been doing a lot here in Chile, you feel very full. I don't know what the language is in Spanish, but it's full is actually a very mechanical term. But all of research is on how much proteins and fats you sense. No one has actually looked at how much stomach stretch contributes to how much you eat, when you stop eating, obesity, diabetes. So these molecules are kind of going to let us study novel areas of biology from the perspective of pressure sensing that no one had thought about before. So again, this was curiosity-driven research, but how does it help people? How does it help patients? One of the interesting things is that before we knew these patients that I told you about, that they don't sense touch and proprioception, the clinicians had completely misdiagnosed them. They thought they had motor neuron problems. They thought they had muscle problems, for example. It's finding that they are touch insensitive by itself is already very helpful to them, because now they know that the eyes can compensate for their lack of proprioception. And before they were telling them, just like you and me, when you walk, to look straight for physical therapy. But now that we know that their proprioception is deficient, it's actually very important for them, unlike us, to keep looking at what their limbs are, because that's the only information they can get. So just knowing is already helpful. But we're also, as I mentioned, developing small molecule blockers of piezo2, and hopefully they'll be useful to treat chronic pain. And I also hinted at this from the beginning. You know, when we first looked at this, we didn't even think about how piezos can be related to systems such as blood pressure sensing, bladder sensing, that could be very beneficial for hypertension, urinary dysfunction, etc. I'm going to close by a couple of slides to tell you that piezos are not just in vertebrates. They are actually, this is an evolutionary tree. So everything I've been talking about, the mammalian piezos are on the left. But piezos are in many other species, including little flies, worms, plants, even unicellular organisms. And we've done one study, for example, on a plant. You would ask, why would plants need to sense pressure? There's actually many reasons. One of them is that roots, when they go into the ground and they experience a harder surface like a rock, they have to detect it and decide to go through it or to go around it. And to do this, the tips of the roots are thought to be mechanosensitive for doing exactly this. So my postdoc found that piezo in blue is exactly expressed, is on where the root tips, where this is happening. And he actually showed that, just like I showed you what the mice without piezos can sense touch, he showed that without piezo, these plant roots cannot penetrate this hard surface, and they come to the surface and go around it. And this visual, the way it's focused, you can see it in the mutants, but not in the normal wild-type plants. It's just a beautiful example to me of how evolution, once it finds something that works well, uses it in a variety of different ways. So the same protein that we use to sense touch is used by plant to sense the hardness of the surface that it has to, the roots has to go through in a plant. I think it's remarkable. So that's all I had today, and thank you for your attention. And I think there's time for questions if anybody wants to ask questions. I'm getting close to the fan for some cooling.
Hi. Thank you so much. That was truly illuminating. I'm just curious to know if the piezos can have scales in a person. Can you have a lot of them or a little? Is there gradients in the piezos in each human being? I'm thinking about my daughter. She never knows when to go pee. I have to constantly remind her. I'm like, well, maybe she has little piezos in her little bladder. Is that possible? ¿Puedo, quieren que diga la pregunta en español? Sí, digo, let me just...
I think someone's translating already.
Perfecto.
Yeah. So I think it's a very good question, and we don't have exact data for that yet, in the sense that, but it's very possible. It's very possible that someone has too much piezo and might feel too much bladder function or too much pain, and someone might have very little and not feel it enough. The way we're addressing this now is there are large databases. One of them is called UK Biobank. So in British people, 500,000 people are sequenced, so they know what kind of piezos and all other genes they have. But they also have all their medical records associated with those people. So now we can go in and search these kind of variations are associated, what we call genotype to phenotype. So phenotype is the sensation, the genotype is what kind of genes they have. So there's so much more to study, but the thing you're saying I'm very interested in, because we can look at different places and see how much of the protein there is. Is there variation? Does that cause differences between humans? Because if you don't have it at all, it's pathogenic, it's a disease. But slight variations could decide between normal variation that we all experience probably in this room.
Thank you. Thank you so much.
Thank you very much. It was wonderful. I was thinking about the role of the membrane, and if you can change the rigidity or fluidity of the membrane, it would change the activity of the channel. Do you have data on that?
I love it. That's a technical science question.
Yeah.
Uh, so he's asking that if they're sitting in this fatty membrane and stretching it opens it, then if you change the property of the membrane so that if it's more fluid or more hard, it should change how easily you can open it or close it. And that's absolutely right. We actually have some data to suggest exactly that. So we know it can. What we're now looking into is if that's how some of the signaling is regulated in animals. Good question.
Thank you.
Hi, thank you. Well, I have a question. It's more related to external conditions, maybe. Is these PSOs can be related to gravity of the planet or maybe the atmospheric pressure, the environment pressure?
Don't know yet. I am considering collaborating with NASA to do some experiments in outer space to see if piezo's affected. Just we looked at it in the plants that I talked about. So the Arabidopsis plants always grow up, right? But because they grow in a petri dish, after they grow up, you can turn it 90 degrees, and the plant actually makes a sharp turn and starts going up again. Without piezo, this was working normally, which means in this case, there must be another sensor for it that we still don't understand. So I think one of the important things to highlight when we give a talk like this is not just to explain what we already, not just to say that what we found explains everything, but to highlight all the things that we do not know yet, and this is one of them. So there must be other forms of gravitational sensing that we have no idea yet.
Thank you.
Sorry. I got a question about piezo. Is the fact, for example, I practice martial arts, and in martial arts, when you about micro fractures in bones, for example, and the bones start getting more stronger. So, how does that work about piezo? Because, for example, if I told you that I get more resistant to pain from when I was 10 to now that I am 19, training that martial art, so the thing is, how does physically or morphologically piezo change about that?
Yeah, you guys are asking great questions. Very much related. Actually, I didn't have time to talk about it, but so for example, it is known that I'll give another example from martial arts. Professional tennis players, their dominant arm has higher bone density than the other arm, which means that the more impact pressure bones sense, the denser, the stronger they become. So there must be a sensor, right,
Yeah.
that is responding to external stimuli. Similarly, when astronauts go into outer space and don't exercise, don't sense gravity at all, they come down, you measure their bone density, and it's very much so lowered. And so that's another example that you constantly need, we are built to sense this gravitational force. So, you know, the tennis player was on one extreme. On the other extreme, if you don't sense anything, your bone density starts going down. So this is not my study, but others have shown that without piezo 1 in the bone density goes lower. So in this case, actually, piezos are playing an important role in sensing the pressure in the bones and regulating how strong the bone gets. For fracture and injury, no one's looked at, but it's possible it's related.
And another small question, since you talked about perception and piezo one, what does happen to piezo when someone has phantom pain or lose a limb?
Yeah. So phantom pain, for those who don't know, so some people who have, for example, lost an arm, it's the worst thing ever because they feel pain, for example, in their fingertips that they don't have. It's one thing not to have it, and to feel pain on it is even worse, right? And there's lots of models about how this works. Many people think that it's a brain problem, so it has nothing to do with what I'm talking about. But it's not completely true,
Yeah.
because if you put anesthetic at the place where it's cut, for example, the phantom limb goes away. So that means that there's signals coming from those cut nerves that probably express piezo that are being misrepresented in the brain. So there probably circuitry has changed in the brain, but the signal's still coming from the periphery. So when we make blockers of piezo2, that's one of the indications we're hoping to test in a clinical trial.
Thank you for your presentation. It was super nice. I would like to know if we born with the perception or we developed while we are raising, like we get in a natural way.
Absolutely, proprioception is learned. I mean, as you know, babies are not coordinated. And so I think as we experience life, we slowly become better at being coordinated. And so this is very important, but we don't know the answer to this. So for patients who don't have piezo2, who are not coordinated, if we are able to repair the piezo gene in the adult, will they be able to have normal functioning proprioception or not? We don't know the answer to that. We're starting to test them in animal models, so hopefully we'll know in the next year or two.
Hello, my name is Eduardo. First, I have a little question for you. If you have this receptor, it's a peripheric receptor, and thinking about the chronic pain. If you have a human with chronic pain, you have some changes in the central nervous system, like a brain. If you put in a topic, a stimulus in the peripheric, in the skin, how you will change these changes in the brain. One thing is nociception, other thing is pain. Pain is a response, nociception is a stimulus. I don't know if you understand my question, but how you will change this brain change with a topic stimulus.
Yeah. So we don't have the tools to do the experiment you're talking about. So we are actually developing both activators and blockers of piezo2 to test questions like that. But we can't do it right now. Um, but I can imagine that if we had such a thing, it would feel like very unusual combination of sensation of touch, proprioception, maybe even pain at the same time. But we just don't know yet.
Thank you.
My question is that you earlier say that it's probable that some people have more of piezo, so there's more people that they are likely like they have higher pain tolerance than other people. So why does that happen? Why there are some people that have a higher pain tolerance than others? Is that because there's more piezo, but why they have more than other people?
We talked a little bit about this from a previous question. That's theoretically possible, but we haven't been able to look at that directly yet, but we will. But as you can imagine, people who are more or less sensitive to pain could be for many reasons. Pain is very complicated. It could be because of peripheral sensing. It could be because in some part of your brain you amplify that signal more than others. You might be really good at suppressing pain. So, it's a very complicated circuitry, and at every node you can control if you feel too little or too much pain. So this is one possibility, but there's many other ways to achieve that.
Hola, buenas tardes. Una consulta. Usted dice que hay sensores térmicos, de presión y químicos. Tenemos también en la piel sensores que puedan detectar energía, por ejemplo, ondas sonoras. La gente sorda puede sentir música a través de la piel, en teoría. O cuando dos personas asecan sus palmas, no se tocan, igual se siente la sensación de otra persona. ¿Tiene que ver con sensores distintos?
I don't know if everybody heard this. The question is, we have temperature sensors, pressure sensors. How about other kinds of sensors? He called it energy, but talked about, for example, the vibrations we feel from loud music or certain frequencies. Many of these actually would go through the piezo channel. So many of the sensors of piezo are actually different frequency sensors. And so vibrations, whether it comes from hands vibrating or music vibrating, if it's very loud, will affect mechanosensors that I talked about. But hearing is different. So hearing is also vibration sensing, but they seem to use different molecules than piezos.